So in this lesson or module, I'm going to talk about introduction to bold fMRI, blood oxygen level dependent fMRI. I would recommend before viewing this, view my introduction to functional imaging methods generally. And I'm going to pick up right from the last handful of slides from that one when we talk about bold fMRI. So as I talked about in the last module regarding functional imaging, MRI and other techniques can give us images of the brain, which can be incredibly helpful to look at neuroanatomy and different parts of the brain. What they don't tell us is about brain function. And these are cross-sectional images. These are sagittal views of a T1 and a T2, which are different types of MRI approaches to look at the brain, spinal cord, and you can even see the base of this individual. And you see the brain, and the black here is the ventricles, and the brain here, and the white is the ventricles. And I won't get into the differences of T1 and T2, but the point is these are just images. They don't tell us anything about brain function. And having the structure for brain images is really important. But just as a reminder from the ENIGMA-U module on the physical principles of MRI, we acquire our data one slice at a time. So each of these is a slide going very quickly through these different views, the axial, sagittal, and coronal. So you can see these are many slices, and we acquire each slice at a time, and we put it together to make a three-dimensional view of the brain. But not only do we acquire the data slice by slice by slice, but we also have information in each of the voxels, or the little cubes in each slice. And I'll show you an image of that as well. And that becomes really important when we talk about functional MRI. And so one issue to address is that you'll sometimes hear individuals say an fMRI machine, or they're wondering if they can use an MRI machine for functional imaging. And the answer is yes. There's no difference between an fMRI and a clinical MRI machine. Both of these images on the bottom here are both structural images, and what we do with functional MRI is just overlay a different signal on top of that. And that different signal we'll talk about, but this gives us an idea of where neurons are firing the most, where based on blood flow. And so there is no difference between an fMRI and a clinical MRI machine. All MRI machines can be used to look at function. So it's a really important relationship to establish before we get into bold fMRI. And that's the relationship between neural firing and blood flow. And so, and if you look at the A, there's less neural activity, and so there's less flow. But then in B, there's more neural activity, and now this the vasculature expands, there's more oxyhemoglobin coming in, and there's greater flow. So there's a direct relationship between neural firing and blood flow that we capitalize on in fMRI. And so the fMRI mechanism looks something like this: increased neural activity, increased blood flow, more deoxygenated blood, less oxygenated blood. And why is that? That's because in areas where neurons fire, oxygen is metabolized, and so there's more deoxygenated blood. Deoxygenated blood goes up. This relative ratio creates an imbalance, and now there's an influx of oxygenated blood, and that increases our bold signal locally. The word locally is very important here. The increased bold signal doesn't happen across the entire brain. It happens where there was more, and just in the regions where there was more neural firing, which gives us an opportunity to track differences across the brain in the bold signal based on how much neuronal firing is happening. And so Sujo Ogawa in the early 90s examined this using an MRI sequence. It looks like a structural brain scan, it's called T2 star, noted that it might retain information about blood flow, which could be very important to examine physiology, and therefore examine brain function using bold fMRI methods. And so if you think back to your ENIGMA-U physical principles of MRI, T2, which is just a type of scan, brain scan, which gives you this image on the left, where the ventricles are white and the darkest areas are white matter, and the gray matter looks gray. A T2 star is a slightly different T2 scan, which has structure, but it's acquired very rapidly, and it's not cleaned up in the same way a T2 is. And because it's acquired rapidly and it's not cleaned up in the same way, it may have information in here. At each voxel and each slice, it retains some information about blood flow. And because of that, it allows us to look at across all of this space where there's more flow and where there's less flow. So when you hear T2 star is a common type of bold fMRI imaging. Again, the exact same scanner. You could run this scan and then stop the scanner. You have this picture, and now set up the sequence slightly different. And now you have T2 star, and these are bold data. So what's in essence happening? Pow, I think about something. So there's more neurons firing here in the prefrontal cortex because I'm doing math or I'm something cognitively demanding. And now we're going to track the relationship between oxygenated blood and deoxygenated blood. And so there's some baseline level of a ratio of this here locally where I was thinking about something. So watch the bold signal here. At first, when the deoxygenated blood increases, my bold signal drops out. So this is the fMRI signal that I'm trying to track. And this is the spatial dimension. This is my voxel. And so this is just a little cube of space that we're acquiring data. We have thousands of these cubes all over the brain that we can look at this signal. Now, because of the imbalance between deoxy and oxyhemoglobin, oxygenated blood begins to flow in. So neurons fire, neurons are firing, oxygenated blood goes up, and now so does the bold signal. And what we can measure then is in areas where there's heightened neural firing, there should be this response and therefore an increase in bold signal. And this is what it looks like. And it's called, there's a fancy word for it. It's called the hemodynamic response function. Hemo, blood, dynamic, changing. So the blood changing response, and they put it into a function so we can see it. And so there's this period of neural activity early on. There's this what we call initial dip, which we don't really focus too much on. But in essence, this is when there's greater deoxyhemoglobin, we lose our signal, and then our signal goes up and it peaks at around 6 seconds and then drops back down. So if we know this, we know that in areas of neural firing, we should get a blood response that looks something like this. We can look at all the voxels across the brain, all of the areas in the brain where there's more blood flow, and it should tell something about neural firing. So a couple of things to know. This blood oxygen level dependent signal, the fMRI signal, is not a measure of oxygen perfusion. It doesn't tell us like PET in a previous module can give us an exact measure of how much oxygen is being used. Bold, it has a number of different parts to it, and it's not just oxygen being used. It's also not a direct measure of neuronal firing, and this is a little confusing. Bold, the blood flow, is directly related to how much the amount of neural firing in an area. However, it is still a measure of flow. It's an approximation of how much flow using MRI techniques. Unlike EEG or MEG, which directly measure the electrophysiological changes related to axonal propagation, this bold is a measure of flow, which is related to neuronal firing. And so we call it a indirect measure of neuronal firing. It also doesn't have an exact quantity, and that can be a little confusing, but in essence, what we look at is change in its properties over time. It doesn't give us an exact number that means anything. We want to look at how much it's changing against its own baseline. What we do know about the bold signal, the fMRI signal, that it's linked to neuronal firing directly. And so it is a reliable, indirect measure of neuronal firing. It has a time and a shape that are reasonably predictable. And I showed you that waveform a moment ago. It usually comes on and peaks at around 6 seconds. And so we can look for that shape, that signal, across the entire brain pretty reliably and know that when we see it, there's more neurons firing. And so now you know a little bit about this T2 star image, which is not very exciting, right? How do we get the pretty pictures then that you'll see in studies of fMRI, where these areas here, the red or the yellow, are areas where there's more neuronal firing than the rest of the brain? In essence, what we're trying to do is, again, I said that when we're going to acquire our data slice by slice, but also cube by cube by cube, voxel by voxel. And so what we want to do is we acquire them quickly so that we can get a whole brain in about 2 seconds. We turn on the scanner, the person's engaged with a task or lying still, and we're acquiring these data slice by slice by slice, and we can get an entire brain, all those slices, all those cubes, in 2 seconds. Gives us an opportunity then to collect a full volume, and then another 2 seconds later, another volume. And so over the course of 10 minutes, you're going to have hundreds of full brain volumes, and each of those full brain volumes has thousands of these voxels that we can look at. It's a lot of data, as you might imagine. How do we analyze it? Well, in the simplest form, really what we want to do is look at the same voxel across all those time points, 1, 2, all the way to the end of time, our time period in the scanner, and we're just going to plot out that bold signal. How much signal do we get? And again, it doesn't have a value necessarily like oxygen perfusion or anything like that, but what it does have is we can plot it out in that voxel against itself to see what's happening to that signal over time, okay? And if it's changing over time in a meaningful way, then we can say, okay, neurons are firing there in a way that's predictable. And if it's not, then we can say it doesn't seem to be much meaningful change. In this case, it looks like this voxel is going on and off cyclically, almost in response to something. And we may have a task, of course, that we have given them, or even if it's at rest, we can track what is going on with each of these voxels and how they relate to one another. So we call that time series data. So every 2 seconds, we get a new volume. We collect our data 1 slice at a time, put all those slices together, done. We have a volume, another volume, another volume. And what we're going to do is, again, look at just all of the voxels, and we're going to look for that bold signal in each of the voxels and model it. And the reason why fMRI works is because if we only had one image, it would just be a picture. It would just be a static image, much like all of the other structural MRI that people are most familiar with. But because we acquire the data very rapidly, it only takes us 2 seconds to create this, we have the opportunity to look at change over time. It becomes dynamic or changing. We have 2 seconds, and then 4 seconds, we have another volume, and we can look at how that signal is changing over time in our time series. And so if you just took voxel A, so this is voxel A, you could plot out how much signal is happening in this voxel over time. And again, this one looks like it's coming on and going on and off, on and off. So then an important question is, what is activation? Or these, if you look at in any study using fMRI, these points of red here that indicate more neural firing. How do we get that, right? And so in its simplest form, those areas of red, or those blobs, represent those voxels or areas showing more bold response compared to any baseline. And so what we might do in the scanner is, if we did the simplest task, would be just, you're lying in the scanner and the scanner starts and you're resting. And then on the screen, it says, tap your fingers. So at first it's off. And then you just tap your forefinger and your thumb together gently during this window of time for on. And then off, you stop. And then on, and then off, you stop. And what we're going to look for then over this timing, so we know what timing this is. You were not tapping your fingers, then you were, then you weren't, then you were. Now we're going to look for, across the entire brain, where were there blood flow changes that seem to match this timing? And so if you look over here, now these are all of the voxels in the brain. There's only 9 here. You'd have thousands of these. And the software helps you analyze these data to look and see, based on this timing, which voxels where the bold signal, the blood flow, seemed to go up and down in sync with your timing. And if it is in sync enough, we would say it's active, right? And so this doesn't, for example, this voxel doesn't look very active. This might be, this starts to look active. These look much better. It's not going to be perfect on, off. But what we're looking for is differences. For example, this voxel versus this voxel. Those are quite different, aren't they? And so for those that are the most different, that stand out the most, we'll call them, if you look up in the image up here, they'll get color-coded as red. And so now we can begin mapping based on some task. You can begin mapping which areas are neurons more likely to be firing during that task than not. In this case, it would be the motor cortex because you're tapping your fingers. One last concept that's important to discuss when you're talking about brain activation, and that is signal-to-noise ratio, or SNR. And SNR is really about the signal that you want to identify in the background of physiological noise. And so what that means is, if we go back to the finger tapping experiment, you're having the participant tap their fingers on, off, on, off. But the brain is also processing other information all the time. There's blood flow everywhere in the brain based on different neural firing patterns that are not related to the finger tapping task. And so what you really want to be able to do is pull your signal, meaningful signal that you're interested in, those areas that are involved in tapping your fingers, beyond the baseline level of flow and what we would call noise. Now, it's not noise to the participant. They need that blood flow and neural firing patterns to live. But what we really want is to isolate only that blood flow that's related to neural firing associated with finger tapping. And so you might imagine, this is just a hypothetical, where there's some true signal that we really want to identify. And then, of course, there's noise, or there's other patterns of blood flow in the brain. And therefore, the observed signal that we get is going to look something like the true signal intermixed with noise. And then we analyze those data in order to pull out what we're hoping is the true signal, but our results are probably going to be intermixed with some noise and some real signal. And the higher the SNR, or signal to noise ratio, the better off we are in order to get the true signal in our results, as opposed to a lot of noise in our results. So in summary, bold fMRI research can measure large-scale changes in brain states, meaning large assemblies of neurons working together and firing together to change blood flow. The signal's based upon flow, so unlike EEG and MEG and some of the other techniques that examine electrophysiology, it's an indirect measure of neural firing. There's a very well-established relationship between neural firing and blood flow. But bold fMRI does not measure neural firing. It measures the flow, the blood flow related to neural firing. Bold fMRI data can be collected on any MRI machine. Brain function is determined by examining that change in signal across thousands of voxels, those little cubes in the brain, over time. So we might acquire 10 to 20 minutes of data in any experiment. What we're looking at in studies of activation, those areas of red or yellow, when you look at brain maps, is a regional or local increase in the bold signal, which indicates greater neural firing in that location compared to other areas. And so where our brain maps are really looking at what is the change in any region based on my task. And you remember the finger tapping task. Is that you're tapping your fingers, you stop. You're tapping your fingers, you stop. As that signal, if areas in the brain follow that timing, that bold signal is oscillating in the same timing, then we will say that is a local increase in the bold signal due to neural firing. It looks like that region is active in relationship to my task.